Welcome to Module 2 of the course Tissue Biochemistry. In the previous module, we explored the biochemical characteristics of muscles, the kidney, the liver, and adipose tissues. In this second module, we will shift our focus to three vital areas. 1. General metabolism of the brain and neuronal biochemistry. 2. Biochemistry of reproductive tissues. 3. Detoxification and excretion in tissues. This module will provide you with an in-depth understanding of how these tissues contribute to human physiology and health at the molecular level. Let's begin with the brain and neuronal biochemistry. The brain is one of the most metabolically active organs in the human body. It accounts for about 20% of the body's total energy consumption despite constituting only about 2% of body weight. 1.1 Energy requirements of the brain. The brain relies almost exclusively on glucose for energy under normal conditions. Unlike other tissues, it has limited energy stores and cannot perform anaerobic metabolism effectively. Thus, it requires a continuous supply of glucose and oxygen. During prolonged fasting, the brain adapts to use ketone bodies, primarily beta-hydroxybutyrate and acetoacetate, produced by the liver from fatty acids. However, this metabolic shift takes several days and is not as efficient as glucose metabolism. 1.2 Glucose transport and utilization. Glucose enters the brain via glucose transporter 1, GLUT1, in the blood-brain barrier and GLUT3 in neurons. Once inside the cells, glucose is metabolized via glycolysis, the tricarboxylic acid, TCA, cycle, and oxidative phosphorylation to generate ATP. Importantly, neurons do not store glycogen, but astrocytes, a type of glial cell, contain small amounts of glycogen and can supply lactate to neurons during metabolic stress. 1.3 Neurotransmitter biochemistry. Neurotransmitters are chemical messengers used by neurons to communicate. They can be classified into amino acid neurotransmitters, glutamate, excitatory, GABA, inhibitory. Biogenic amines, dopamine, norepinephrine, serotonin, histamine. Neuropeptides, substance P, endorphins. Acetylcholine, involved in muscle activation and memory. Each neurotransmitter has a unique biosynthetic pathway. For example, dopamine is synthesized from tyrosine via tyrosine hydroxylase and dopa-decarboxylase. Serotonin is derived from tryptophan. GABA is synthesized from glutamate via glutamate decarboxylase. 1.4 Myelin biochemistry. Myelin is a lipid-rich sheet surrounding axons that facilitates rapid nerve impulse conduction. It is composed of lipids, 70 to 80%, and proteins, 20 to 30%. The major lipids include cholesterol, sphingomyelin, and cerebrocytes. Oligodendrocytes produce myelin in the central nervous system, while Schwann cells do so in the peripheral nervous system. 1.5 Brain biochemistry in disease. Many neurological disorders have a biochemical basis. Parkinson's disease, loss of dopamine-producing neurons. Alzheimer's disease, accumulation of beta-amyloid and tau proteins. Epilepsy, imbalance in excitatory and inhibitory neurotransmitters. Multiple sclerosis, autoimmune destruction of myelin. Underscore 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 underscore. 2. Biochemistry of reproductive tissues. Reproductive tissues undergo complex biochemical processes, particularly during development, sexual maturation, pregnancy, and lactation. 2.1 Male reproductive biochemistry. The major male reproductive organs include the testes, epididymis, vas deferens, and accessory glands such as the prostate and seminal vesicles. Leydig cells in the testes produce testosterone, the principal male sex hormone, from cholesterol via a series of enzymatic reactions involving cholesterol side chain cleavage enzyme, P450-SCC. 17-alpha-hydroxylase. 17,20-lyase. Testosterone is crucial for spermatogenesis. Development of secondary sexual characteristics. Libido and muscle mass regulation. Sertoli cells support spermatogenesis and produce inhibin, which regulates FSH secretion from the anterior pituitary. 2.2 Female reproductive biochemistry. The primary female reproductive organs include the ovaries, fallopian tubes, uterus, and mammary glands. The ovary produces the steroid hormones estrogen and progesterone. These hormones are derived from cholesterol and play key roles in regulation of the menstrual cycle, development of secondary sexual characteristics, preparation and maintenance of pregnancy. Estrogens are synthesized via the aromatase enzyme, which converts androgens to estrogens. Progesterone, secreted by the corpus luteum, prepares the endometrium for implantation and supports pregnancy. 2.3 Biochemical changes in pregnancy. During pregnancy, there is a dramatic increase in the levels of human chorionic gonadotropin (HCG), progesterone, estrogen, and placental lactogen. The placenta acts as a temporary endocrine organ, producing HCG, maintains the corpus luteum in early pregnancy. Progesterone maintains uterine lining. Estrogens promote uterine growth and blood flow. Relaxin relaxes the pelvis and cervix. 2.4 Biochemistry of lactation. Prolactin stimulates milk production, while oxytocin facilitates milk ejection. Breast milk contains proteins, casein, lactalbumin, immunoglobulins, lipids, triglycerides, cholesterol, essential fatty acids, carbohydrates, lactose, which is synthesized from glucose and galactose. Underscore 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 underscore. 3. Detoxification and excretion in tissues. Detoxification is the biochemical modification of potentially harmful substances to facilitate their excretion. This process occurs primarily in the liver, but other tissues like the kidney, lungs, and intestines also contribute. 3.1 Phases of detoxification. Detoxification is classically divided into Phase 1, Phase 2, and Phase 3 reactions. Phase 1 reactions, functionalization. These involve oxidation, reduction, or hydrolysis to introduce or expose functional groups. The cytochrome P450 enzyme system is the main catalyst. Examples: oxidation of benzene to phenol, hydrolysis of esters. Phase 2 reactions, conjugation. These add polar groups to enhance solubility. Common conjugates include glucuronic acid via UDP glucuronosyltransferase, sulfate via sulfotransferases, glutathione via glutathione's transferase. Phase 3 reactions, transport. Modified compounds are actively transported out of cells via ATP-binding cassette, ABC, transporters for excretion through bile or urine. 3.2 Organs of detoxification and excretion. Liver, primary site for drug metabolism and detoxification. Kidney, filters waste products such as urea, uric acid, creatinine, and drugs. Lungs, excrete volatile compounds like alcohol and anesthetics. Skin and sweat glands, minor route of excretion for certain salts and urea. 3.3 Metabolic waste products. Urea, result of amino acid catabolism via the urea cycle. Uric acid, end product of purine metabolism. Creatinine, from creatine phosphate breakdown in muscles. Bilirubin, from heme breakdown. Abnormalities in their levels indicate underlying disorders. For example, elevated urea and creatinine suggest kidney dysfunction. High bilirubin indicates liver or hemolytic disease. 3.4 Xenobiotics and biotransformation. Xenobiotics are foreign compounds such as drugs, pollutants, and toxins. They undergo biotransformation to become less toxic and more excretable. The liver's microsomal enzymes handle most of this transformation. However, sometimes toxic intermediates are produced, such as acetaminophen metabolites that can cause liver damage if glutathione is depleted. 3.5 Antioxidant defense systems. To combat the harmful effects of detoxification byproducts like reactive oxygen species, ROS, tissues have evolved antioxidant systems including Superoxide dismutase, SOD. Catalase. Glutathione peroxidase. Vitamin C and E. These enzymes neutralize free radicals and protect tissues from oxidative stress. Underscore 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 underscore. Module 2 Summary. Let's briefly recap the key points discussed in this module. The brain depends heavily on glucose and oxygen for its function, and neurotransmitters play a key role in neuronal communication. Reproductive tissues are hormonally active and undergo profound biochemical changes during puberty, pregnancy, and lactation. Detoxification is a multi-step biochemical process involving transformation, conjugation, and excretion of toxic substances primarily via the liver and kidneys. Various antioxidants and enzyme systems help the body deal with metabolic byproducts and environmental toxins. Underscore 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 underscore. Course conclusion. Congratulations! You have now completed both modules of the tissue biochemistry course. You have learned how specialized tissues such as muscle, kidney, liver, adipose tissue, brain, and reproductive organs function at the biochemical level. How energy metabolism varies among different tissues and how hormones regulate these processes. How tissues contribute to detoxification, waste removal, and maintaining homeostasis. This knowledge is crucial for understanding disease mechanisms, clinical diagnostics, and therapeutic interventions in modern healthcare and biomedical research.